that we will handle this morning is implantation and placental development. But prior to that, we will talk about the ovarian and endometrial cycle. Anyway, the objectives will be to correlate the hypothalamic, pituitary, and gonadal secretions with ovarian gonadal synthesis. To understand the two-cell gonadotrophin theory of ovarian steroid production, to know the effects of estrogen and progesterone on the endometrium, and to explain and understand why a woman menstruates. Now, let me talk first about the ovarian cycle and the endometrial cycle as an introduction. You know that cyclical ovulation continues during almost 40 years between menarche, which is the onset of menstruation, and menopause, which is 1 year cessation of menstruation. So there are 400 opportunities in the lifetime of a woman that she can be pregnant. And this capability to get pregnant is tightly regulated by the hypothalamic pituitary ovarian axis. So we will try to understand this, that the cyclical changes in the lining of the endometrium or the endometrial histology are faithfully reproduced. And these are the hormones that are contributors to the process in the changes of the endometrium from a proliferative to a hyperplastic endometrium. And the hormones most involved are the gonadotropin releasing hormones from the hypothalamic area, the luteinizing hormone from the pituitary gland, as well as the follicle stimulating hormone, and of course, the ovarian production of estrogen and progesterone in response to the hypothalamic pituitary ovarian stimulation. So the average duration of the endometrial and ovarian cycle is 28 days plus or minus 7 days. And this is the average interval also for women who are regularly menstruating. In the 28-day cycle, plus or minus 7 days, the follicular or proliferative period can be variable, can be prolonged, but the luteal or the post-ovulatory phase is constant at 14 days. So if your menstrual cycle is prolonged, what is prolonged is the proliferative phase, meaning follicular phase in the ovary, proliferative phase in the endometrium, because there's constancy in the duration of the luteal phase in the ovarian cycle and the secretory phase in the endometrial cycle. Now you have had this in physiology, and so this is but a review for you. As you can see here, this is a developing antral follicle. You can see here that these hormones will be, these ovarian cells, the granulosa cells, will be producing estradiol, and the latter part of the cycle, progesterone, okay? Now, under the influence of your hypothalamic releasing hormone and your pituitary gonadotropins, your follicle stimulating hormone stimulates the follicular phase of the ovarian cycle. Once ovulation occurs, the luteinizing hormone now stimulates and maintains the life of the corpus luteum. And a well-developed corpus luteum, therefore, has this level of estrogen and progesterone, meaning the secondary in estrogen and the high levels of progesterone. After a set number of days of 9-11 days, the corpus luteum lyses, okay? But before it's under the effect of your estradiol, the endometrium goes into a proliferative phase. And after ovulation, when the corpus luteum secretes progesterone, you now have the secretory phase of the endometrial cycle. When the corpus luteum lyses and there is a drop in estrogen and progesterone, there is collapse of the endometrial surface leading to menstruation. And with collapse of the corpus luteum, there is no more inhibition on the pituitary secretion of FSH and luteinizing hormone, and a new cycle begins. That's how we correlate our hypothalamic, pituitary, ovarian, and endometrial cycle. So let me go more in details. You can see here, as illustrated topographically, the ovarian cycle. As I said, we have the follicular phase, ovulation, which is the release of the egg. This is the follicular phase of the ovarian cycle, where the primary oocytes, primary follicles develop into the mature Graafian follicle. When the mature Graafian follicle releases the egg, ovulation took place, and the Graafian follicle becomes the corpus luteum cyst after release of the egg and secretes now your progesterone and estrogen. But the lifespan of the corpus luteum is 9 to 11 days. So it lyses, and when it lyses, there is trapping estrogen, progesterone, and there is no more inhibition on the brain production of FSH and LH, and a new cycle begins. If you can see here, the appearance of a Graafian follicle, you can see here the granulosa cell layer and the external theca layer, the antrum, and you have the oocyte within the cumulus or operus. Okay, and immediately surrounding the oocyte will be your corona radiata. This is cumulus or operus, and this is the corona radiata, and that is the egg that is to be released during ovulation. Now, let us look at prepubertal life, okay? In the embryonic period of the female, the first 6 weeks, you have primordial germ cells. This is in utero, okay? The first 6 weeks of ovarian development, primordial germ cells migrate to the genital ridge, okay? There is rapid mitosis, and the first meiotic division occurs. So at 20 to 24 weeks, there is the first meiotic division. The primary oocytes are in meiotic arrest, and they are arrested in the prophase of the first meiotic division. Okay, so at birth, you can have about 2 million oocytes. Okay, actually at life, you have about 6 million primordial germ cells. But at birth, there are only 2 million oocytes. Most of them have the fate of atresia. At puberty, only 400,000 of these oocytes resume its development and undergoes resumption of the meiotic division, of the first meiotic division. In the reproductive lifespan of a woman, about 400 oocytes are matured and be capable of being fertilized. So at birth, we have 2 million oocytes. At puberty, 400,000 oocytes. And at birth, we have 400 oocytes. Okay? The rate of depletion of follicle per month until 35 years old is 1,000 follicles per month. So you can see here that women who are trying to get pregnant after the age of 35 years may suffer from relative infertility because her ovarian reserve is poor. The percentage of follicles which underwent atresia during lifetime is 99.9% of all the follicles. And as I said earlier, during the reproductive life, 400 follicles with its egg during the entire reproductive age. So let me now go into detail on the stages of human follicular development, the ovarian cycle. The follicular phase is composed of the preantral phase, and the preantral phase is gonadotropin independent. The growth of the follicle here comes from the oocyte. And then later on, you have the antral stage, which is gonadotropin dependent. Okay, so preantral stage is gonadotropin independent. The antral stage is gonadotropin dependent. So when the mature Graafian follicle is fully mature, ovulation takes place to release the egg. And the Graafian follicle is transformed from a granulosa theca layer to a granulosa lutein and theca lutein cell, which is called the luteal phase of the ovarian cycle. This is a fully developed follicle in the follicular phase of ovarian development. As you can see here, you have the oocyte within the cumulus or ovarus, surrounded by a layer of corona radiata. This is antral fluid secreted by the granulosa cells, and lining the follicle is the granulosa cell layer. External to the granulosa cell layer is the fibrous tissue cellular theca layer. So this is the theca interna, the theca externa, the granulosa cell layer, the antral follicle, the antrum, and then you have the mature follicle, mature oocyte surrounded by the corona radiata and the cumulus or operus. What causes resumption of meiosis of the fully grown oocyte? Remember that at birth, the oocyte is arrested in its first meiotic division. The surge of luteinizing hormone coming from the anterior pituitary gland initiates resumption of the meiosis of the fully grown oocyte. Okay. So looking now at this particular discussion of mine, we look now at the follicular development of each follicle. And now we look at the two-cell, two-gonadotropin principle of ovarian steroid production. This is the theca cell layer. The theca cell layer secretes androstenedione, testosterone, and cholesterol. The source of cholesterol synthesis by the theca cell is the circulating luteinizing hormone. Okay. The luteinizing hormone is responsible for the development of the theca cell, and the source of cholesterol, my mistake, is the LDL, low density lipoprotein, coming from the systemic circulation. And then this is converted inside the theca cell into androstenedione. The androstenedione now goes into the granulosa cell layer. In the follicular stage of follicular development, there is a basement membrane that separates the granulosa and the theca cell layer. Once ovulation takes place, the basement membrane disappears, and there is free passage of blood between the theca cell and the granulosa cell. So androstenedione is now transferred via diffusion to the granulosa cell. And the aromatase system in the granulosa cell, under the influence of your follicle stimulating hormone, converts androstenedione to estradiol and is then released into the general or systemic circulation. So under the two-cell, two-gonadotropin principle of ovarian steroid hormone production, the theca cell produces androstenedione and testosterone. The granulosa cell secretes estradiol, estrone, and a very important hormone that is secreted by the granulosa cell is inhibin. Inhibin exerts a negative feedback to the pituitary gland to inhibit the FSH release, resulting to non-development of other follicles, enabling only one follicle to reach maturity, and that is the mature Graafian follicle. So the inhibin has an effect on a negative feedback to the pituitary, so the pituitary gives lesser FSH secretion, so only one mature Graafian follicle develops. So let me now go to follicular development of the ovarian cycle under the influence of follicle stimulating hormone. Actually, the rise in follicle stimulating hormone comes in the luteal phase of the previous cycle. So in the luteal phase of the previous cycle, there is already an increase in FSH receptors in the preantral follicle. Okay, this enabled aromatization of the theca cell-derived androstenedione into estradiol. Okay, so FSH rises from the previous luteal phase, causing stimulation of release of estrogen. Estrogen responsible for the aromatization of the theca cell-derived androstenedione, which is secreted under the influence of the luteinizing hormone. The two-cell, two-gonadotropin theory. Okay, now how about luteinizing hormone coming from the anterior pituitary gland? The luteinizing hormone receptors appear in the pre-ovulatory granulosa cells begin to secrete small amounts of progesterone. Progesterone exert a positive feedback on the estrogen-primed pituitary. So the minimal progesterone secreted by the granulosa cells exert a positive feedback effect on the estrogen-primed pituitary to cause the pituitary gland to release luteinizing hormone. So you will now be able to determine more or less when the woman will ovulate, because estradiol peak occurs 34 to 36 hours before ovulation. Estradiol peak occurs 34 to 36 hours before ovulation, whereas your luteinizing hormone peak occurs 10 to 12 hours before ovulation. It is the estradiol peak that stimulates the anterior pituitary gland to secrete the peak of luteinizing hormone, and this peak occurs 10 to 12 hours prior to ovulation. So, when the mature Graafian follicle matures, rather ruptures to release the egg, it is because there is an LH surge coming from the anterior pituitary gland. Okay? The LH surge initiates resumption of meiosis of the fully grown oocyte. It proceeds from the diprotein state of meiosis 1, which was started during fetal life. Ito, hindi ba sabi ko sa inyo kanina? Oogenesis, primary oocyte, development of the follicle, as you can see here, undergoing the first meiotic division. The first meiotic division is completed after puberty, and the secondary oocyte is released. The secondary oocyte is picked up by the fallopian tube and fertilized, and meiotic division starts. Okay? So you can see here a mature gamete with the ovum inside, and you have here the second polar body. Okay? So repeat. The LH surge occurs 34 to 36 hours before ovulation. Estradiol surge occurs 10 to 12 hours before ovulation. And the oocyte now proceeds from the diprotein stage of meiosis 1, which was started during fetal life, to metaphase meiosis 2. At ovulation, meiosis is again arrested, and meiotic division is only taking place if that oocyte is fertilized. I repeat, at ovulation, meiosis is again arrested, and this will only be completed upon fertilization. What happens during ovulation? It's the process of follicular development, okay? As you can see here, this is the fallopian tube, this is the ovary, and this is the mature oocyte that is about to be released and taken by the fimbria of the fallopian tube. There is augmentation of the cumulus oophorus, expansion of the cumulus oophorus surrounding the oocyte. There is remodeling of the extracellular matrix, and there is weakening of the follicular basement membrane. So proteases weakens the site of the follicle where the ovum is supposed to be released. So I repeat, release of the mature oocyte to the surface epithelium is called ovulation. This is brought about by augmentation and expansion of the cumulus oophorus, remodeling of the ovarian matrix induced by luteinizing hormone, and weakening of the follicular basement membrane brought about by the proteases and also the prostaglandins. So you can see here, this is an illustration of the development of the follicle. As you can see here, you have your Graafian follicle. This is the selection, recruitment. This is the mature Graafian follicle. Ovulation has taken place. After ovulation, the Graafian follicle becomes the corpus luteum. There's a breakdown of the basement membrane separating the granulosa and the thicalosal cells, and there is vascularization of the granulosa cell, because before ovulation, the granulosa cell is avascular. After ovulation, blood vessels and their capillaries invades the granulosa cell layer. So there is vascularization of the granulosa layer. and there is hypertrophy of the granulosa cells with increased capacity to synthesize hormones, mainly progesterone and also estrogen. What brings about the expansion of the oocyte of the follicle? What brings about the expansion of the follicle? It's a process that happens during the production of the hyaluron-rich extracellular matrix. The cumulus cells surrounding the oocyte becomes loose in contact with one another, and they move towards and outward from the oocyte. It results in augmentation of the cumulus complex volume. So lumalang ang expansion is because of increased production of your extracellular matrix and augmentation of the cumulus complex volume. There are substances that influence the genes of the cumulus complex to form the hyaluron-rich extracellular matrix. So what are these hormones that bring about, or growth factor that bring about a hyaluron-rich extracellular matrix? Luteinizing hormone from the pituitary gland, progesterone from the granulosa cells, prostaglandins from the cumulus ovarus, and the growth development factor 9, growth differentiation factor 9, and the bone morphogenetic protein, which comes from the oocyte. So I repeat, luteinizing hormone from the pituitary, progesterone from the granulosa cells, prostaglandins from the cumulus cells, and your growth differentiation factor and bone morphogenetic factor coming from the oocyte. Okay? And these are all factors that cause the maturation of the Graafian follicle and its eventual rupture. So after rupture of the mature Graafian follicle, what happens now to the follicle? That is what we call luteinization. We are now in the luteal phase of the ovarian cycle. The first was follicular phase. The second is the luteal phase, which takes place after ovulation. The granulosa cells hypertrophy with increased capacity to synthesize hormones, estradiol. At day 2 post-ovulation, the basement membrane separating granulosa and thicker lutein cells break down, and there is neovascularization of the granulosa in response to vascular endothelial growth factor produced by the granulosa lutein and thicker lutein cells. There is hypertrophy of the granulosa lutein and thickest lutein cells. Mas malaki yung nakikita nyo, there is no more basement membrane between granulosa lutein and thicker lutein. It's richly vascularized because of vascular endothelial growth factor produced by the lutein cells and the hypertrophy of the granulosa lutein and the thicker lutein cells. Let me now describe the luteal phase. The corpus luteum after ovulation is formed. It has a greater capacity for granulosa lutein cells to produce progesterone, as you can see here, okay? From the systemic circulation, cholesterol, androstenedione aromatized to become estradiol. Cholesterol is aromatized to become progesterone. So it comes also from cholesterol, okay? And all of this, progesterone and estradiol, enters the systemic circulation. Due to enhanced access to blood-borne, low-density lipoprotein-derived cholesterol, which is the estrogenic precursor. The precursor of synthesis of androstenedione and progesterone is LDL-derived cholesterol coming from the systemic circulation. So if we are going to describe the luteal phase, what are the hormones produced by corpus luteum? Okay, the thicker lutein cell produce androstenedione and testosterone. The granulosa lutein cells produce estradiol, estrone, progesterone, and the peak of progesterone rise is at 20 to 50 milligrams per day occurring in the mid-luteal phase. Yun ang pinamataas na level ng progesterone nyo. If the woman does not get pregnant, the corpus luteum collapses. When the corpus luteum collapses, estrogen and progesterone levels drop. There is no more inhibition on the anterior pituitary gland, and a new cycle begins. However, if the woman gets pregnant, the human chorionic gonadotrophin released by the trophoblast of pregnancy maintains the corpus luteum. There is endocrinologic similarity between luteinizing hormone and human chorionic gonadotrophin because both act via the same luteinizing hormone, human chorionic gonadotrophin receptor. So again, this is the two-cell gonadotropin principle of ovarian steroid hormone production. In the follicular phase, you have here follicle stimulating hormone stimulating the granulosa cells to produce its estrogen, as you can see here. But in the follicular phase, also the theca lutein cell stimulated by the luteinizing hormone converts cholesterol into androstenedione. The androstenedione goes to the granulosa layer and is aromatized into estradiol. Okay. Now, in the luteal phase, the theca lutein cell has a lot, because it's highly vascularized, has a lot of cholesterol, which is also converted into androsterone, which can be released directly into the circulation, or it may enter the theca cell layer, converted and aromatized into estradiol. And progesterone coming from the cholesterol also goes to the systemic circulation, the two-cell, two-gonadotrophin theory. Any questions so far?
None po.
Okay. So we now go to the luteal phase of the ovarian follicle. Okay, the luteal phase. Tapos na tayo sa follicular phase of the ovarian cycle. The lutein cells produce progesterone. And the granulosa lutein cells, it's not only the theca lutein cells, but also the granulosa lutein cells that secrete progesterone. And the production peaks at 25 to 50 milligram per day at the mid-luteal phase. So at the peak of production of progesterone by the corpus luteum, you can assay 25 to 50 milligrams per day. So how about the estrogen production during the luteal phase? There is a secondary rise in estrogen production during the luteal phase. Of course, it is decreased after ovulation. The estradiol drops after ovulation, but it is followed by a secondary rise at the mid-luteal phase. Ito, okay? And ang estradiol, ayan ang progesterone. And the amount is 0.25 milligrams per day. The secondary decrease in estradiol production lasts until the end of the cycle. So sabay bumabagsak ang progesterone at estradiol level. So we are now in the luteal phase of follicular development. The Graafian follicle becomes the corpus luteum, which is a transient endocrine organ with a lifespan of 9 to 11 days after ovulation, and then undergoes apoptosis. The mechanism is unclear, but believed to result, the lysis of the corpus luteum is believed to result from decreased LH levels and decreased LH sensitivity of the luteal cells. So after ovulation, decreased levels of LH and decreased sensitivity of the luteal cells to the action of luteinizing hormone. Therefore, that phase is characterized by the dramatic drop in circulating levels of estradiol and progesterone, resulting in the collapse of the menstrual endometrium, and the woman menstruates. Let us talk first about the ovarian hormones. The most biologically potent, naturally occurring estrogen is 17-beta estradiol. It is very important for regulation of follicular development. Remember, the 17-beta estradiol is secreted by the granulosa cells. It regulates follicular development, the Graafian follicle, and it regulates uterine receptivity and blood flow in the avascular follicle. It has 2 steroid hormones produced. It has 2 intracellular receptors, estrogen receptor alpha and estrogen receptor beta. These ER complexes act as transcriptional factors via the estrogen response element. How about progesterone? Again, it's a steroid hormone. There are two receptors like estrogen, two intracellular receptors, progesterone receptor A and progesterone receptor B. There is also secreted a G protein associated progesterone receptor, but the action of this is unclear. So what is the biologic response of the progesterone receptors? The progesterone receptor complexes act as transcriptional factors via the progesterone response element. When these receptors are co-expressed, progesterone receptor alpha A inhibit progesterone receptor B gene regulation. So progesterone receptor A inhibit progesterone receptor B gene regulation. Also, progesterone can also increase intracellular calcium. I have finished the ovarian cycle. Do you have any question? Any question on the regulation of the ovarian cycle?
None po, Doc.
So shall we proceed now to the endometrial cycle? As I said, you are always free to interrupt me in case there is some misunderstanding on what I am saying. Okay. We now go to the endometrial cycle. I repeat, the hypothalamic pituitary ovarian axis regulates the changes in the endometrium of the uterus. Okay? Hypothalamic releasing factors act on the pituitary gland to release your FSH and your LH, which is responsible for changes in the ovar follicular phase and the luteal phase of the ovarian cycle. The follicular phase secrete estrogen, the luteal phase secrete progesterone. And you can see now that in the follicular phase of the ovarian cycle, there will be progressive growth of the lining of the endometrium, which is so-called the proliferative phase. Okay. When the ovulation and follicle ruptures occur and develops into a corpus luteum, progesterone is produced. Thus, you have the luteal phase causing the secretory phase of the endometrial cycle. When the corpus luteum collapses after 9 to 11 days and there is no pregnancy to maintain the corpus luteum collapses, estrogen and progesterone diminishes, the endometrium collapses, and a new cycle begins because the inhibition of estrogen and progesterone on FSH is removed. Okay? So when menstruation occurs, the endometrium is left away when the corpus luteum regenerates and a new cycle begins. Okay? So let me look at the endometrium. The endometrium is formed by the epithelial lining, okay, the glands, the spiral arterioles, and the stroma. This is the basement, stratum basalis, and with the myometrium. This is the uterine artery branches, the arcuate artery, the radial artery, the basal artery, and the spiral arterioles. What participates in the menstrual cycle are the spiral arterioles. So you have the epithelial lining in the glands, you have your epithelial lining in the stroma, you have the spiral arteries and the stroma. There are two layers in the endometrium, the functionalis layer and the basalis. The functionalis layer participates in the changes during the endometrial cycle and is shed off during menstruation, whereas the basal layer, which contains the basal artery, is the source of regeneration of the endometrium and blood vessels. As mentioned here, functionalis layer is shed and is reconstructed from the basal layer of the endometrium. So what do you mean by recovery of the endometrium? It is important that the endometrium recover so that there is cessation of endometrial bleeding. And the cessation of endometrial bleeding is brought about by reepithelialization and angiogenesis. So there is formation of new epithelium and formation of new blood vessels. This is dependent on tissue regrowth, which is estrogen mediated involving both estrogen receptor A and estrogen receptor B. The production of estradiol in the follicular phase of the ovarian cycle is the most important factor in endometrial recovery after menstruation. I repeat, the estradiol production in the follicular phase of the ovarian cycle is the most important factor in endometrial recovery after menstruation. So let us look at how the endometrium recovery after menstrual blood and its superficial endometrium is shed off. Epithelialization of the superficial endometrium begins before menstrual bleeding stop. Nagdudugo pa siya, nagre-regla pa siya, nagre-epithelialize na yung endometrium. And by the 5th day of the menstrual cycle, epithelialization is completed already in the shed-off endometrium. And what is responsible for this epithelialization are the epithelial cell growth factors. The epithelial cell growth factors are estrogen growth factor, epidermal growth factor, transforming growth factor A. So these are the growth factors that are responsible for epithelial growth after menstruation. Estrogen growth factor, and the transforming growth transforming factor, which is responsible for regenesis or regeneration of the endometrium. As far as the stroma is concerned, the stromal cell proliferation is enhanced and favored by estrogen, your fibroblast growth factor. So by the 5th day, naka-recover na yung lining endometrium nyo that has been shed during menstruation. The formation of blood vessels, which we call angiogenesis, is brought about by estrogen raising the level of vascular endothelial growth factors. So let me discuss the endometrial cycle. The early phase is the proliferative, and second half is the secretory phase. In the early proliferative phase of the endometrial cycle, you can see that the endometrial glands are straight and narrow. Okay, they follow a parallel course towards the superficial layer of the endometrium. From the base, they assume a straight course towards the endometrial cavity. Okay, on day 5 of the menstrual cycle, you will see mitosis in the glandular epithelium. Day 5 of menstruation. So you have here your spiral arterioles, your spiral vein, and your endometrial glands, and you have the stroma. Okay. In the late proliferative phase of the endometrium, what do you notice? There is glandular hyperplasia. There's still mitosis. There is increase in stromal gland substance, and there is edema and proteinaceous material in the stroma surrounding the glands that are developing. In the functionalis layer, the stroma is loose and the glands are widely separated. But in the basal portion, the stroma is dense and the glands are more crowded. So in the late proliferative endometrium, which is nearing ovulation, day 14 in a 28-day cycle, the endometrium is already thick. As you can see here, the glands are tortuous, the spiral arterioles are coiled, there is a lot of stroma, as you can see here. The surface epithelium acquire microvilli, which causes an increased epithelial surface area, and they develop cilia, which aid in the movement of endometrial secretion from the glands towards the lumen of the uterine cavity. In this phase of the endometrial cycle, the glandular epithelium are tall and pseudo-stratified. Progesterone can only act on an estrogen-primed endometrium. Kahit marami kang progesterone, pag walang estrogen, your endometrial changes will not occur. because a secretory and endometrial progesterone responds to an estrogen-primed endometrium, or estrogen-primed endometrium responds to a rising progesterone in a particular manner. So we go now to the secretory phase. The first sign of ovulation is seen histologically by the presence of glycogen vacuoles, okay, in the basal portion of the glandular epithelium. You can see the glandular vacuoles in the basal portion, in the basal portion of the glandular epithelium. By day 18, the vacuoles go towards the surface of the gland, to the apical portion. So from its apical, day 17, from its basal or subnuclear, day 17, day 18, it's in the apex of the glands. And by day 19, you will now see glandular secretion in the lumen of the glands. And these glandular secretions contain mucopolysaccharides and glycoprotein. Remember that mitosis in the gland occurs only in the proliferative phase. Day 5 and onwards, you have mitosis of the glandular epithelium during the proliferative phase. But once there is a secretory endometrium, glandular mitosis ceases because of the anti-mitotic property of progesterone. Please remember that. Glandular epithelial mitosis ceases with secretory activity because of the anti-mitotic effect of progesterone. And at this point in time, your 17-beta hydroxydehydrogenase converts your estradiol to a weaker estrone. So we now go to day 21 to 24, and your stroma is very edematous, your glands are tortuous, your spiral arterioles are highly coiled. Okay, this is day 22 to 25 after ovulation. The striking phase endometrium undergoes striking changes. The predecidual cells or the stromal cells surrounding the spiral arterioles become enlarged and become the predecidua. Stromal cells surrounding the spiral arterioles enlarge, and stromal mitosis becomes apparent. So in the secretory phase, there is stromal mitosis but no glandular mitosis. The glands become extensively coiled and secretion becomes visible in the lumina of the glands. When is the best time for a fertilized egg to implant in the endometrium? Remember that in a 28-day cycle, fertilization takes place on day 14 along the fallopian tube. So when is the best time for that fertilized egg to implant in the endometrium? The window of implantation is between 20 to 24 days. Why? The surface epithelium has fewer microvilli and cilia. The pinopods living with protrusions on the apical cell drives the egg towards the fertilized egg towards the endometrium. And it helps, the pinopods help the endometrium prepare for blastocyst implantation. And the glycolycs allow acceptance of the blastocyst. So this is the mechanism of implantation. Surface epithelium develop microvilli, pinopods appear on the apical cells of the endometrial gland, and this helps the endometrium prepare for blastocyst implantation. And the glycolycs in the endometrium allow acceptance of the blastocyst. So you can see here, menstruation, proliferation, secretion, and the implantation window, which is day 20 and 24, the best time for that fertilized egg to implant in the endometrium. Well, if there is no pregnancy, on day 3 to 28, you will see that predecidual cells surround the spiral arterioles. The predecidual transformation of the upper two-thirds of the functionalis layer occurs between day 22 and 25. At this point in time, with the predecidual transformation of the upper two-thirds of the functionalis, you will see extensive coiling of the endometrial glands with luminal secretions, such as secretory endometrium, ready to receive a fertilized egg. So you can see here, as we described earlier, the distribution of blood, no? You have here, this is the placenta, this is the uterus, okay? You have the uterine artery, you have the arcuate artery, the radial artery, and the spiral arterioles, okay? This is the placenta if the woman gets pregnant. So you can see here how coiled the spiral arteries are in the secretory phase and how coiled the glands are. Secretory spiral arteries lengthen at a rate greater than the rate of endometrial tissue thickening. So mas malaki ang coiling at saka length ng spiral arterioles than the rate of endometrial tissue thickening. So the rapid angiogenesis of these arteries is due to estrogen and progesterone-regulated vascular endothelial growth factor. The growth discordance causes great coiling of the spiral arterioles, causing ischemia on the more superficial surface of the endometrium. And so what happens now for menstruation, and this is the third part of my lecture. Now, please remember that in the mid-luteal secretory phase of the endometrial cycle, there is a point when you recognize that the endometrium can receive a fertilized egg. If the woman is pregnant, the corpus luteum is rescued. There will be continued progesterone secretion. The decidualization process continues. Let me explain that. The fertilized egg secretes syncytiothropoblast. Okay, the syncytiothropoblast secretes estrogen, okay, and progesterone. So it maintains the corpus luteum of pregnancy. So it does not undergo luteolysis because it is rescued by the human chorionic gonadotropin coming from the pregnancy that is beginning. So the corpus luteum is rescued. It continues to secrete progesterone. The endometrium does not collapse, but goes into further decidualization. Okay, that is if she's not pregnant. If the woman is not pregnant, progesterone production decreases with luteolysis kasi ang lifespan ng corpus luteum is only 9 to 11 days. And when it undergoes luteolysis, there is no more estrogen, progesterone, the endometrium collapses and the patient menstruates. So how do you characterize the late premenstrual phase endometrium? Characteristic of the late premenstrual phase of the endometrium is infiltration of the stroma by polymorphonuclear leukocytes, giving the endometrial surface a pseudo-inflammatory appearance. This infiltration of polynuclear cells is brought about in the endometrium by your monocyte chemotactic protein and in the endometrial stroma and epithelial cells caused by interleukin 8. So the presence of interleukin 8 and your monocyte chemotactic protein is responsible for attracting inflammatory cells into the superficial layer of the endometrium, giving it a pseudo-inflammatory appearance. The leukocyte infiltration is key to endometrial extracellular matrix breakdown. Matrix metalloproteases and endometrial proteases cause the breakdown of the endometrium extracellular tissue and is also responsible for repair of the functional layer if the woman did not get pregnant. So these are the changes now in the, particularly in the spiral arterioles, as you can see. But on the first day of menstruation, proliferation already begins, characterized by lengthening of the spiral arterioles. I'm talking mainly of the changes in the spiral arterioles. In the proliferative phase, there is lengthening of the spiral arterioles. In the mid-secretory phase, there is extensive coiling of the spiral arterioles. And in the late secretory phase, there is excessive coiling of spiral arteries. So the superficial surface of the endometrium undergoes hypoxia, vasodilatation, and bleeding. So by day 24, tissue volume decreases. Day 25 and 26, severe coiling of the spiral arterioles with blood stasis. Day 27 to 28, hypoxic superficial endometrium with vasodilatation of the spiral arterioles, followed by vasoconstriction and it limits bleeding. The vasoconstriction is the important event following menstruation. So what is there in the secretory endometrium? The secretory endometrium is rich in prostaglandins F2 alpha, and prostaglandin F2 alpha causes vasoconstriction, myometrial contractions, and upregulation of inflammatory responses. Because of its effect on muscular contraction, it can produce menstrual pain. So the ischemia of the muscles as well as the myometrial contraction explains the dysmenorrhea that women feel when menstruating. What is the role of progesterone withdrawal? There is increased cyclooxygenase 2 or COX-2, which is prostaglandin synthase 2. The decrease in 15 hydroxyprostaglandin dehydrogenase degrades prostaglandins. Menstruation is a mixture of arterial and venous bleeding, but arterial bleeding is quantitatively greater than venous. What happens? Why does the woman bleed during menstruation? The endometrial bleeding begins by rupture of a coiled artery with consequent formation of a hematoma, breakdown of the ischemic superficial endometrium, and release of blood. Let me talk in the last few minutes the decidua. The decidua is a highly modified endometrium of pregnancy. It is essential for hemochorial presentation where maternal blood contacts the trophoblast. So maternal blood contacts trophoblast, which is fetal tissue. And so there must be a protective mechanism by which trophoblasts do not invade maternal blood. And that is a function also of the decidua. The transformation of proliferating endometrial stromal cells into specialized secretory cells is called decidualization. Decidualization is dependent on the presence of estrogen, progesterone, and factors secreted by the implanting blastocyst during trophoblast invasion. So estrogen, progesterone, and implanting blastocyst growth factor. So the decidua is a special relationship between the decidua and the trophoblast, ensuring the success of a pregnancy. The decidua produces factors that regulate endometrial receptivity, modulate immune and vascular cell functions with the maternal fetal environment. Look at the layers of the decidua in a pregnant uterus. Okay, this is the product of conception. This is the decidua capsularis that covers the implanting embryo. This is the decidua This is the decidua basalis, where there is less trophoblast. This is the embryo with the yolk sac. This is the decidua parietalis, which is the less of the uterine cavity. This is the decidua capsularis, which directly overlies the implanting embryo. And you can see, therefore, the decidua basalis is richly infiltrated by trophoblastic cells. The decidua capsularis surrounds the developing embryo, but later in pregnancy, it fuses with the decidua vera. So it is now called this, the decidua basalis is where you find trophoblast and big blood vessels. The decidua capsularis overlies the product of conception and is near the uterine cavity. After the 14th week of pregnancy, 14th to the 16th week, the decidua capsularis fuses with the decidua parietalis so that there is no more uterine cavity. The decidua capsularis fuses with the decidua parietalis and the uterine cavity is no more. There are three layers to the decidua, basalis and parietalis, the zona compacta, zona spongiosa, and zona batalis. The functional layer is still the zona compacta and the zona spongiosa, similar to a non-pregnant endometrium. Predecidual changes in the mid-luteal phase in the endometrial cells appear in the stroma. Okay? These predecidual cells, which we call the stromal cells that enlarge and surround the spiral arterioles. They enlarge to form polygonal or round decidual cells. The nuclei are round and vesicular. The cytoplasm is clear, slightly basophilic, and surrounded by translucent membrane. A decidual reaction in the stroma is completed only when there is blastocyst implantation. So pre-decidual muna, but once blastocyst implants, then you have a full decidual reaction. So what is the fate of the spiral arterioles? Along the decidua capsularis, the fetal blood vessels, the spiral arteries are lost. Okay? In the decidua parietalis, it persists because it lines the entire uterine cavity, and the decidua basalis is markedly altered and invaded by the trophoblast. Walls of the decidua basalis are destroyed by the cytotrophoblast. This is where the fetus and the placenta become, the decidua basalis and the trophoblast becomes part of the placenta. The zona espongiosa in early pregnancy composed of large distended hyperplastic glands with minimal stroma. As pregnancy advances, the glands disappear. The decidua basalis contains a zone of fibrinoid degeneration, aka Nitabox layer, and it is believed that this is a protective mechanism against trophoblastic invasion deep into the muscular layer of the uterus. The cells of the decidua are the decidual cells, maternal bone marrow derived cells, lymphocytes, which are regulatory T cells, decidual macrophages, and decidual natural killer cells. They are found in the uterus. As you can see, the decidual natural killer cells, they have a role in the immunity reaction of the implanting blastocyst to a foreign maternal environment. The accumulation of lymphocytes at the maternal-fetal interface is essential to invoke tolerance mechanisms that prevent maternal immune rejection of the fetus. See, these are protective cells so that the blastocyst can implant and not be rejected by the maternal tissue. The decidua is the source of enormous amounts of amniotic fluid protein, although its exact physiologic role is unknown. The accumulation of lymphocytes at the maternal-fetal interface is essential to evoke tolerance mechanisms that prevent maternal immune rejection of the fetus. Please remember, this is a significant role of lymphocytes at the maternal-fetal interface because that is essential so that the maternal organism can evoke tolerance mechanism and will not prevent evoke tolerance mechanism that will prevent maternal immune rejection of the fetus. So with that, I am done with my lecture. I have a few minutes left, I think. I have 7 minutes for questions and answers.